So thank you everyone for joining us today. We're gonna have our first NPOmix workshop in English. We had a workshop in Portuguese in February. So it's my pleasure to be here today. And it's my pleasure to have here today with us Assistant Professor Justin van der Hoft from Wageningen University in Netherlands. And he's gonna give us a brief introduction on the topic. Justin, the floor is yours.
Okay, Tiago, you will have to allow me to share my screen.
Sure. One second. I should have made you a co-host. um,
Well, in the meantime, let me say hi to all of you. I'm glad I see so many of you. Some names are recognized, some names not. So, well, welcome. Um, my, my pleasure meeting you all. Um, you should see something now. And for those of you that have been to Florence, you may even recognize it. Am I right?
we don't see
And
the
then,
we see a cathedral.
yes,
Now
it's
this
actually the Dome of Florence. But now you should see my slide. Yeah?
yes.
Okay, cool.
Yes.
So, okay, welcome. So thanks, Tiago. Thanks for organizing the workshop. I think it's a great initiative, and I hope it's the start of many more to come on integrated omics mining. So we thought it would be nice to give some background motivation and some information about the concepts that undermine integrated omics mining. So I'm Justin. I'm working in Wageningen. So I started my own group there 2 years ago. So on the picture you can see that it's growing. So we have 3 PhD students working with me, and Joris, assistant research assistant, and he's actually working on integrated omics tools, so enhancing MP linker, and I will mention that tool later during this short introduction. So I also would like to stress that most of the work that we will discuss today, including NPOmix, it's building on a lot of previous work by a lot of others. These are the various labs that I was working over the past 10 years. So, and especially today, in view of the topic, Peter Dorstein, as we mentioned, as a key driver and motivator of this type of integrative omics work. And also would like to stress that through funding, for example, from the eScience Center, a lot of the tools and resources that also you hear today have been established. So most research starts with a very simple question. We see these molecules. You may wonder who's producing them, and what happens? Do they make the plant happy or sad? If we talk about plant microbiomes, or do they make us happy or sad if we talk about ourselves? However, that question is usually not as simple. And I'm coming here from the metabolomics point of view, but you could equally come from the genomics point of view, where you would look at genes or unannotated genes or groups of genes. And here we look at spectra that are, it's not the same as structures. So the first question, whether you have genomics or metabolomics data, will always be, what is in there? And then we can start to ask questions like, what is it doing? So I think most tools, including genomics ones, metabolomic ones, they rely on natural patterns to be found. And you can see here that humans have been inspired by patterns from nature for years. So this church they started to build this in the 11th century, and already then they were fascinated by patterns from nature. Flowers, you can recognize them in the roof to keep this structure up and alive, and it's still there today. So, and the motivation to really mine bacterial and plant genomes for interesting specialized metabolites are manifold. I just wanted to include some of them here, like the climate crisis we are in, and the need for enhanced plants that can withstand the drought and the salt stress that is inevitably coming in many regions of the planet. And that will also be important to keep the yield and nutritional value of our staple crops, at a good level. And I think most, if not all, of these specialized metabolites somehow impact growth and health of plants, but also of ourselves. So they are often key, and finding out who is producing which molecules and what the structures are and what they are doing is key to a lot of natural product related research. So why are we here is because there are challenges in this type of research that are still unsolved, unmet, and but we all know that plants and fungi, bacteria, fungi, plants, they produce a myriad of molecules that are very interesting. Yeah, not only for themselves but also for us. You can see some examples on the slide, but the challenge there is that whether we look at mass spectra or at gene clusters, is the large-scale coupling of genomic or spectral data to molecular structures of the known ones even already, and especially the novel natural product molecules. And just to highlight that especially the larger molecules is where we have the hugest trouble with, because there adding a few heavy atoms to this, for example, vancomycin structure, already gives a kind of combinatorial explosion of possible structural configurations that we then have to solve or reduce to something that we can actually test or link in terms of structural elucidation. So there is really a need to do combined genome and metabolome mining. Why? Because then we can actually make direct links between the structures and the producers. And if we can do that, then through the genome, where we can now start to recognize these biosynthesis gene clusters, we can also look around the gene clusters and look for functional genes, and genes are annotated, for example, resistance related genes. And we can also make a link to the possible functions of these molecules, but also on a more kind of or less functional level for the structural elucidation. It can be very helpful to have information from the genome available to us. For example, in peptides, using mass spectrometry, we can start to identify the type of amino acids that are linked to each other, but often the stereochemistry is hard, if not impossible, to find. And there the gene cluster information may help us if we see particular epimerases present in the gene cluster or not. So in other words, this combined genome and metabolome mining helps us to link molecules to the producers, and also it can really help us to obtain structural information in a complementary manner. So as I already started to say with the picture of the church, pattern mining, natural pattern mining is really a key asset, right? So, and you can do that either supervised or unsupervised. And Tiago will tell you a lot more about how the NPOmix approach works. And I would say that's more of an example of a bit more supervised way of finding patterns. But in any way, pattern mining is a really natural thing to do. And I'm confident that some of you already saw this picture, but for those of you that did not see the picture, you still have seen already within a few seconds why I took this picture, right? Two groups of animals, in this case, easy to distinguish by the human eye. Our problem is that the groups of genes and the groups of spectra are not so easy to distinguish by the human eye. So there we need this computational genomics and computational metabolomics tools to help us. And as you can see here, I provide examples of finding molecular families based on the spectral similarity and extract building blocks of metabolomics based on substructures, right? But equally well, you can go to the genome side of things if that's where you're starting from. You can start to find the gene clusters and group them using tools that are now available like Bigscape and BigSlice into families or clans of gene clusters, all producing similar or related, structure related molecules. And also, and this is ongoing work with Marnix Medema, one of the anti-smash developers, and Joris Lauwe, the research assistant I mentioned, we can start to find these building blocks, these substructures also from the gene clusters. So hopefully more on that later this year. And if we can use that information and link that across, then we can really start to exploit this pattern mining at a higher level. So within genomes or within metabolomics, we can already use it to propagate structural features and to annotate family chemistries, for example. Yeah. So within the currently ongoing NPOmix project, together with the eScience Center, one of the most prominent solutions is the paired omics data platform, and I'm confident Tiago will also come back to this. So there's a platform that we established together with a lot of contributors. I saw some more names in the audience today to record paired omics data. And it sounds simple, but in practice, it was very hard to find this paired omics data sets, as we had to dig into the literature, into the PDF supplements, and then somewhere, if you were lucky, there was a table where the actual links were mentioned. Now it is present in this resource. It's computer readable, and it's a lot more easy to reuse. I'm not saying it's a perfect solution. Still, you will see that some genomes may be hard or difficult to download, or there's other things that are still not perfect, but at least I think it's a huge step forward in making this data, this paired data, more available to researchers like Tiago who want to develop tools to improve the linking. So what it does is it brings together the sequence genomes and the LC-MS data. Sequence genomes, for example, in CPI, and the LC-MS data, for example, in metabolites or GPS mass. But not only that, you can also add validated links between gene clusters and molecular families or molecules based on the spectra. So, and that is also very important to do if you have a data set. Please contribute, right? But not only that, if you can add the validated gene clusters to the paired omics data platform record, that will be really helpful for tool developers like Tiago and myself and others, because we can use them as a validation of the new tools. So the URL of the paired omics data platform is provided on top. So have a look around if you are interested in this kind of work. And yeah, so to add your own data, it works pretty straightforward. So there was a whole team of more than 100 researchers in the end that contributed, and with a core team of 15 people, we decided on what was the minimum metadata that we needed. So yeah, these are examples of what you would need to input, but we want to make it as lightweight as possible without giving up on the effective reuse of the data, basically. So, and more information on that is also in the paper that is cited on the slide. And then when we come to the actual linking step, that's here where we have LC-MS URL to the metabolite or massive entry of the file itself, and then you can add the genome label and all the experimental steps that were used to come to the metabolomics data set. I wanted to highlight that actually very recently, if you would happen to have a proteomics data set of the same sample, then you can even now also edit within the paired omics data platform. The first example is already available on the paired omics data platform from the University of Leiden. And also on the right-hand side, you can see what we ask in terms for the BGC versus MS links. You can already see that we link out to MyBig. MyBig, for those of you that don't know it yet, it's a database of validated gene clusters, and it's a very important resource for us because that is where our ground truth is. So if you have a curated gene cluster, your data, the first step would be to add it to MyBig, and then you can also link it out in the paired omics data platform. It's a win-win. You have more exposure, and also the community will be able to reuse your data more effectively. Yeah. So and else we ask for some structural information like a smiles or inching, so that we can actually also assure which structure we talk about. So for those of you that are really genomic oriented, I noticed in the slides of Thiago that some of you are really coming from the genomic side. That's fine. I have some slides on what is a mass spectrum that Thiago and myself mostly work with. So the mass spectrometer typically gets a lot of ions in, focuses them, and then is able to differentiate and separate them based on the mass over charge. And we even get some sort of relative abundance. And that is what is a mass spectrum in any type of, from any type of instrument. And most instrument types nowadays are also able to do an additional step that is key to our workflows, and that is the fragmentation step, where a bit of energy is added, usually in the form of an inert gas like argon, and then the ions, the fragile ions, are then fragmented, and then the so-called MS2 or MS-MS or tandem mass spectrum emerges. It's the same setup of the previous mass spectrum, but now we look at fragments. And if you wonder about all these names, in my opinion, they all mean the same thing, but I think some people will be able to tell you why it should be called tandem mass spectrum, and others why it should be called MS2 spectrum. That's all fine, but in the end, they all mean something like this. Okay, so and it's this MS spectra that most computation with genomics tools nowadays work with, like molecular networking, MS2LDA substrate discovery, and also NPOmix. So there's a nice bridge to molecular networking, not completely unexpected. So as a key tool that emerged over the last 5 years, available from the GMPS platform. And this has the goal to organize and group your mass spectra. And why I mention this, because it is it can be used as input for integrated omics mining tools that not only the spectrum itself, but also the molecular family membership. And it's important to realize that this is done based on spectral similarity. When we zoom out, and I fully agree that this is quite a complicated slide, but yeah, as I hope you already had realized that this whole challenge is quite complex. So here we can actually see and appreciate how everything works together. And if you want to study it more with more time, then the reference cited on the slide also has this figure and more information on it. So on the left-hand side, we have the genome mining side. Yeah. So another shout out to tools like Antismash, spearheaded by Minus Medema and Tilman Weber, and the MyBig database as well, that are heavily kind of really propelled the genome mining field. And now the clustering algorithms, Bigscape, and the latest kit on the block is a Big Slice that can do the clustering on a larger scale. That was Satya Kautsa's work, and really it can create a lot of gene cluster families, and they are also often used as input for integrated omics mining, gene cluster family membership. And then on the right hand side, we have the tools that I already briefly skimmed over now from the metabolome mining side. So molecular networking from GPS, MS2LDA substructure discovery. And also, I think it's good to highlight and mention that we can also start to predict compound classes based on spectra, right? We have this nice ontologies available now, a classifier, MP classifier, that are able to automatically, based on smiles, classify molecules, and we have the tools that are able to add through in silico approaches the kind of the candidate molecules or smiles, or directly the class ontologies like through tools like Canopus. So in the end, that means that for each feature in the data or molecular family in the data, we can have a predicted compound class. And that is important to realize, because from the genome mining side, we also get sometimes such predictions on whether it's a peptide, whether it's a photoketide, etc., etc. So we can use that information, and actually, if you're interested, Joris Lauwe and Marnix Medema, we just have a preprint out that actually shows how you can use this chemical compound class predictions to further, to remove a lot of false positive links from your data. And I think Tiago will also highlight this in his part of the workshop, of his talk. So yeah, that brings me to what are then the key concepts? Where does NPOmix fit in? Well, right here, right? So on top of NP linker. So it kind of capitalizes on the paired data that's available in the paired omics data platform, and it then aims to bring together the gene clusters on the left-hand side and the spectra, the mass spectra on the right-hand side. And it does that using a supervised approach that I will leave to Tiago to fully explain. What I will do is I will briefly indicate that there's an alternative approach around, like the NP linker framework that was published last year, that also is able to use as input data from the paired omics data platform. It then grabs the data. You can add, or it will produce so-called user data, so annotations like Bigscape is run, molecular network can be run, and that information is then combined, and lists of ranked lists of VGC, MSMS spectral links are produced. And in the paper, and the reason I show this is that now again, I would like to stress not only to add your data to the paired omics data platform, but also to add this so-called red dots in this plot, because each red dot here is a validated BTC MSMS link, and was used to assess how well a novel score or how complementary two scores were. So really, it is really important that we keep collecting these examples. Yeah? And again, a big shout out to all these important resources like MyBig, validated gene clusters, NP Atlas, structural information on microbial molecules, and mass bank with high quality mass spectral data. So all the references are also on the slide if this is new to you. So yeah, the future of natural product discovery. Yeah, so this is a slide that I grabbed from the IOMICA project that is integrated omics for metabolomics and genomics annotation. And I'm glad to say that as of next month, the NP Linker 2.0 project will start, also together with the eScience Center, where we will further develop and upgrade NP Linker, the NP Linker framework, to make it more user-friendly and to add enhancements like visualization and such. But here I would like to indicate how would it look like. Yeah, so on the left-hand side, we see the gene cluster families. On the right-hand side, we see metabolite families or molecular families. And the idea then would be to have this combination of strain correlation-based matching, which the Metcalfe score is an example. Metabologenomics, they term that. So basically, you look at a gene cluster family, which strains, which microbes produced it, and then you look at the family, which strains are present in the molecular family, and then you look at the overlap, and you assign a score based on that. And also the feature-based matching. The feature-based matching can be used to complement that. And what does mean feature-based matching? Well, there we look at chemical classes that are annotated, or substructures, and then we try to see where is a good match between the gene cluster and a spectrum, and or its predicted structure. So yeah, are network products too big to handle? Well, they are very complex, and I think that there's no need to say that that is an easy job. But they're certainly not too big to fail. I think we see more and more examples of how computational tools are able to grab that, get a hold on them, get a handle on them. And I wanted to leave you with some inspiration and kind of how we could also look at things differently. So some of you may know of my work on mass spectral embeddings. So where we put each spectrum in a 200-dimensional world. And usually in the papers you see the 2D images of it, but of course 3 dimensions you can also reduce to 3D. And then together with the Wonder team in Wageningen, we created this tool where you can actually in 3D dive into this mass spectral embedding. And you can even select spectra, the dots, and see their structure and read some information about them. And how cool would it be if in 10 years we could do this for a combined genome metabolome world, where then the gene clusters would be sitting next to the spectra here, and you can actually create the links. So with that kind of outlook and inspiration, I hope you're fully warmed up for Tiago's part now. So thanks a lot for your attention. If you're interested in my work, you can follow my group or myself on Twitter or LinkedIn, and I'm happy to reach out to you or connect. If yeah, and please submit your data to the NPOmix data platform. Thanks.
Thank you very much, Justin. That was a really nice presentation, very beautiful slides. And also you fill some gaps on my presentation that I was not gonna go into the details, and then you explained very beautifully that too. Thank you so much. That was really nice.
Are there any quick questions at this stage?
Oh yeah, nice. That'd be great.
Because I now can easily go back and show the slide again.
Eva raised her hand. You can unmute yourself and talk if you like.
Hello. Hi, Justin. Thank you so much for the presentation. I just have a very quick question. What is the difference between NP-linker and NPOmix?
Yeah, so that's a very good question. I think NP-linker as a framework is really meant to basically do a systematic kind of unsupervised integrated omics mining. And for example, in the future, it could well be that NPOmix will be used, will be run within NP-linker, whereas NPOmix is, yeah, Tiago will explain all about it, is kind of not as unsupervised, and the other way around will not work. Let's put it this way. Yeah.
Got it. Cool. Thank you.
Yeah, exactly. I feel like there's a lot of synergy between both of them, and most likely in the future they'll be working together very nicely. So so we can say that they kind of complement each other, and two different approaches to come up with the same solution.
So another way of saying it would be that NP-linker itself, it uses it's built on other kind of concepts, right? So the whole genomics idea of doing this strain correlation based matching that was not founded by NP-linker. It's founded by Metcalfe et al. and Kate Duncan with the pattern-based genome mining, and those papers introduced that into the scientific literature, and then here we adopted that, and actually there's this standardized Metcalfe score. So in that sense, we built on it, and I think, well, you have to read the paper and decide for yourself, but we improved on the results by standardizing for the size of the gene cluster family and the metabolomic family, and the molecular family.
Oh, just to complement, there is also the training aspect that is slightly different between the two tools.
Yeah, so in fact, there is no training as such in NP-linker, right? So and also you could also if you want, well, in the future, I hope it will be possible to integrate kind of more targeted integrated programs as well, like NERPA. I think you will mention it, Thiago.
Yeah, I'll talk about it very
Yeah.
briefly.
So you could imagine that a program like NERPA that is, I think, focusing on peptides, that the results can be integrated into the results that NP-linker outputs. Other questions? Okay, well, if you still have a question, but somehow I'm not comfortable asking it here, then that's fine. Just send me an email or send me a tweet, and I will find some time to answer you. Okay, thanks.
Yeah, if you also remind yourself of a question later on, you can post in the chat and we can answer at the end. Okay, can you see my screen?
Yep.
Nice. All right, so I'll talk today a little bit about NPOmix, which is a supervised fingerprint-based strategy. By supervised, we mean that we need the algorithm to be trained needs to have labels. So the labels we're going to use are the families that Bigscape generates for the gene clusters. So the gene cluster families. So those are the kind of labels. And then we make the presence kind of a presence absence, but it's more of a similarity absence score for the different strains. And that's the fingerprint. That's the fingerprint aspect. And that's all to connect the natural product mass spectrometry fragmentation data with the biosynthetic gene clusters. So very briefly, just the agenda of the workshop, I'll introduce NPOmix. Then I will show the website, the different databases that we use, and we also suggest that you can submit your data there, and the different inputs. Then one of the inputs is the fragmentation spectra, and so I'm going to show you how to get the fragmentation spectra for the query metabolites, how to explore the NPOmix outputs. And then I'll introduce you, so Justin talk a little bit about the classes, substructures. Those are kind of new features that we're implementing in the version 2.0. We're gonna have a separate workshop on that version with more details in the future, but I will just introduce the version 2.0 today. And also I have a very brief comparison with some other multi-omics tools, still ongoing comparison. So I'll just show the results I have so far. But for example, I'm still comparing NP-linker with NPOmix. So I'll just show you some preliminary results. So it was nice to see that we have a range of different jobs of people that are attending that registered for the workshop. We had 55, and today was actually 58 people registered to the workshop. And most of them are grad students or postdocs or research slash scientists, but we have some others like professors and directors and so on. From many different countries. It's nice to see a large representation of US, but also a lot of people from Germany. Brazil, it makes sense we have some, but not that many because we had a Portuguese workshop last month. So we had a bunch of people, we had 70 people on the workshop last month. So a lot of people interested from Brazil. That's nice to see because I'm from Brazil. So it's nice to see people from my land interested in genome mining, and then so on. But it's also, I think this is one of the most interesting piece of data. Most people are interested on linking genomics with metabolomics, and that's nice to see because it's a new trend. Before people were focused in metabolomics or just genomics, but now it seems that we are trying to connect the dots. So that's why we're here today. So it's really nice to see this kind of interest. So just to introduce NPOmix very briefly, First, Justin did a great job talking about most of the topics that I'll bring it up today. But just to emphasize, there are some things like why is it important to find new natural products? Well, because most of the drugs, they come from natural products or are derived from them. Very few are synthetic or vaccines. So it's nice to find new natural products. But it's good to highlight that the great majority of them were not found using genetics. So there are two ways mainly to find natural products. Like I said, it's either genomics or metabolomics. But they have pros and cons. For example, in this recent study in bioarchives, I think it was not, Justin can correct me if I'm wrong, but I think it wasn't published yet, but the preprint is out. It was a study only bacteria, but several gene clusters, over 1 million gene clusters from bacteria. So it's a very large number of gene clusters. They use big slice to calculate similarity between them. And they estimate, it's just an estimate, but it gives us an idea that only 3% of the potential that we isolated so far can be connected to gene clusters. So there are many, many gene clusters there that probably are not known. I say probably because the moment that we can start connecting them a little bit better and better, we can see more links being formed. And then maybe this estimate changes a little bit. But 3% is a pretty low estimate. That's good for one side. That means that we have a lot of work to do. So that's nice. We're not going to lose our jobs anytime soon. But it's also a little bit challenging to figure out why we're not connecting that many gene clusters to their metabolites. In the bright side, it's more, metabolomics is more of a direct discovery because you get the bacteria, you make the extract, the crude extract, you get interested in a metabolite either because you fractionate it and tested bioactivity, or maybe you did on-target metabolomics and found there's a new MSMS2 fragmentation spectrum in your sample, and then you can go there and try to isolate. Of course, it sounds very simple. It's not that simple. It can take months, maybe sometimes even years, but it's a little bit more straightforward than genomics. Genomics, I say, is a little bit more challenging because most of the time you need to activate those genes, and sometimes it's challenging to activate those genes, or you need to transfer to another bacteria that is more easy to manipulate in the lab. But it can happen that you don't know what metabolites you're looking for. So sometimes it's hard to figure out if you successfully transfer the genes or not. But in theory, you can access the full potential of the gene clusters. So that there's a pro side. So yeah, so they have pros and cons. But the question is, what if we combine them? Can we have a high percentage of the potential being accessed, but also a more direct discovery? So yes, there are solutions for that. There are 8 solutions since 2014. So just very quickly, I really suggest, I have the references in here. I emailed the slides. I will email again the slides after the meeting, but you can check the suggestions more thoroughly, the references more thoroughly to read a little bit more about those tools. They are very interesting approaches, very interesting new technologies that were developed to connect genomics with metabolomics. And sometimes genomics, for example, Gardet connect BGC with SMILES. So it's kind of challenging to call metabolomics, but it's still connecting different different approaches, genomics and structures. So in 2014, the first tool was NRP Quest for NRPSs, non-ribosomal peptide synthetase, and then it connects BGCs with LCMS data. Just very briefly, it tries to predict the LCMS data, and then it makes molecular families to replicate them, and then also see the relationship between them, and then connect them with the BGCs. So that's the major idea. So GNP in 2015, it's for all classes, again, to connect BGCs and LCMS. And now we are dealing with the structure predictions to connect the gene clusters. So Garlic, like I said, is only to connect PGCs with SMILES, not LCMS data. So it doesn't use mass spec, but it can make the prediction of the monomers using genomics and metabolomics, and then try to connect them together. 2019, MetaMiner, a paper from Mohamani lab. I was also part of this paper. We used some bacteria from the Gerwick lab. We were able to connect some RIPs to genomics using this strategy here, trying to predict the monomers and the post-translational modifications, and then make a peptide spectra match that gave us a p-value, and then we can kind of evaluate the quality of the link. So MetaMiner, but it's only for ribosomally encoded and post-translated modified peptides. So is DeepRip, but this time uses machine learning to learn the data, the monomers, the input, and then try to predict the links. So that was more recently 2020. And again, it's only, oh, sorry, I apologize for the color supposed to be red, but, oh, actually it's for both, my mistake. It's for connecting, if you don't have mass spectrometry data, no problem. DeepRip still works, but if you have mass spectrometry data, you can connect also mass spectrometry data to BGCs. So it can both work for both SMILES or OMS data. And then NP-Linker, I'm going to skip because Justin already gave a nice introduction about NP-Linker, but just to mention that it was in 2021 and it's for all classes again. So far, only GNP and NP-Linker are for all classes, so they're not targeted approaches that Justin mentioned. They're more untargeted. They can work for many different classes. And then you can use a mass spec data, but it also uses a little bit of the structured predictions, like he said, with the features that can be used for making correlations. So the last two tools, so I introduced 6. The last two tools are in 2021 as well, just like NP-Linker, NPMiner, but again, it's only for non-ribosomal peptides. So again, try to link based on the predicted assembly lines, so the monomers that it can assemble them, and it tries to generate different backbones and then make the predictions. And at the end, it has a nice network of the different mass spec data and their predictions. And then last tool I would like to mention is NERPA. It's also for NRPSs, but also hybrid NRPSs, PKSs. It does not work for only PKSs, just like Garlic does. So this one has to have a peptidic portion of the molecule. But again, it breaks into monomers. They have the monomer graph, and then identify the monomers from the BGC, and then it tries to make the connections, and then you have those reports with the quality score for the connection. So so those are the solutions, and what we found, and to be honest, most of the solutions are pretty nice. They're very interesting, very good ideas, but the one that works the best so far that I've tested, it's NP-Linker. And we felt really the need of a solution that was systematic, like NP-Linker works for all classes, just like GNP, but the other solutions are not. They are only for NRPSs or RIPs. That was high throughput, like a repository scale. Some of them, for example, DeepRip, online, I wasn't able to find the offline version. I have some links in the end of the slide, but the online version, you can only submit one sample at a time, so it's not high throughput. So we wanted a fast solution because not everyone have supercomputers or servers that they can use the tool. So something fast would be very interesting, like it's offered in the metabolomics world, like Sirius, that it works pretty fast in your computer, so in a regular laptop. So we want something fast and highly efficient. So we want the precision to be fairly high, so we can get more of the true positives and less of the false positives. And that's why we try to create this solution called NPOmix. So just some few terms, we use k-nearest neighbors. That's the kind of algorithm, it's a machine learning algorithm. It's not a deep learning algorithm. It doesn't have like layers and layers. It's actually called lazy learning, which is a funny name for algorithm, but it's basically trying to say that it doesn't try to uh, approximate things and simplify a little bit things. They use the whole data as it is to make the training and then the predictions. So in deep learning, you have nodes that learn a little bit the aspects of the data, the patterns in the data, and then you make the predictions. So that there is this different aspect that you don't use the full data. First, you learn from it, making assumptions, and then you make the predictions. So we are using biosynthetic gene clusters. So again, we're going to use a lot this abbreviation BGC, so biosynthetic gene clusters. And then we're going to group them in families using Bigscape. So Justin briefly mentioned Bigscape. So it's basically looking for the similarity between the BGCs and then grouping them in families that we call GCFs. So here's how it looks, a training matrix from NPOmix. So basically you have here 3 BGCs. It can be many more. I'm just exemplifying with 3. And then just 3 different bacteria. It can be fungi, plants, algae, but I'm just exemplifying here with 3 different bacteria. And it can be many more. For example, in our paper, we're using 1,040 instead of only 3. But just to simplify with 3, it's a little bit easier to visualize the 3D space. So each one of the bacteria, the columns, would be an axis in the 3D space. I'm going to show in a second. But basically, what's this table is showing us is that BGC1, it's absent in bacteria A. And it's present there is something very similar to BGC1 in bacteria B. So 100% similar, bacterium B, sorry. 100% similar to BGC1, which in BGC2 and in the sample in the bacterium B, it's 95% similar. So it's pretty similar, but it's not identical. And that's what this table is showing us. And then BGC3 is absent in B, but it's present in A and in C, and it has a little bit lower score. So it's something more homologous than actually identical. So this is how we plot the axis of A, B, and C, which will be X, Y, and Z in space. And then we can use these icons here, which are representing the GCFs. So BGC1 and 2 have the same GCF circle, and BGC3 is pretty different. It's present in different bacteria, has the triangle, and then that's how we plot them in space. So we can see that, okay, 1 and 2 are actually similar to each other. They're close to each other. And then 3 is pretty different. It's pretty far apart. So just to expand a little bit this example, so just again, the number of samples is equivalent to the number of dimensions. Here I'm using only 3D, but it can be 1,040, for example. That's what we use in the paper. So again, here I added an extra string, and then you can see here the training matrix, which is very similar, but instead of BGCs, we have MS. So, and then we have the similarity scores. Those are similarity scores from Bigscape here in the left side. And then the right side, there are similarity scores, the cosine, modified cosine score from GMPS. So once we have all those scores, we can plot this different BGCs. For example, each shape is one BGC. You can see that the circle BGCs are similar to each other. They're close to each other. So here with this K in space, we are simplifying. Let's say we have 1,000 samples, 1,000 dimensions. We can simplify this multi-dimensional space in only 2 dimensions. That's what we are doing right now. But we can see that the circles are still close to each other. The squares are close to each other, and then so on and so forth. Now we plot the axes, which are the MS data. So the X1 is the first MS spectrum, and we can see that it's close to the circles. So most likely the circle GCF or the circle BGCs are making this X1. So that's how we can connect genomics with metabolomics. And then here in X2, most likely is the square, but there's also a circle candidate. So we can have different candidates according to the number of nearest neighbors, the KNN, the K nearest neighbors, which here is only 3, which is a pretty good number, but it can be more, it can be less. It depends on the user input. So like Justin mentioned, the Pairomics Data Platform is an excellent initiative because it puts together genomics and metabolomics data that can be used to the development of new tools. For example, training this KNN algorithm, and then we use 1,040 paired samples, paired because they have genomics and metabolomics, and this is about over 3,300 BGCs. For testing, we use 11 metabolites and their analogs, a total of 22 MS spectra. We're able to annotate 44% of this spectra, which is pretty good annotation rate. To compare a little bit like GMPS, in the beginning, it had a much lower annotation rate. Now it's expanding and it's getting much better. And the precision rate was pretty good too, almost 80%. So it's an excellent precision for a rather small number of samples. For example, ideally, it would be nice to have maybe 10,000 pair samples. That would be a pretty good number to estimate a little bit better the precision of the tool, and also to use as a database for new samples, new bacteria or fungi or plants. So just to show one example, we had this MS spectrum that it was identified as putative orphamide C. Here's the structure for orphamide C that we think that the MS spectrum is making or is from. And we can see that the amino acids here, then we automatically connected using NPOmix to the orphamide C BGC, which is shown here. The genes are here, 6 genes. And you can see also in these squares, these rectangles, you can see the amino acid predictions. So it makes 9 predictions. The 10th prediction is unknown, but the 9 predictions match in the correct metabolite, the correct amino acid, and in the right order. So it's very likely that this is actually orphamide C. By the mass difference, we can predict that this last metabolite, this last amino acid, sorry, it's actually valine. So we're able to automatically connect. And like Justin said, we can use substructures to reinforce this prediction. Just to emphasize, NPOmix version 1.0 that we use to connect those two does not use the amino acid predictions in the training process yet. So that's something we want to add. So just to finish this introduction, and then I can show you a little bit more the hands-on part, how to submit your samples and prepare them. I just want to say why we're connecting genomics with metabolomics. Well, in the beginning, I said that there are pros and cons from both sides, and connecting them will help to use the more pros than the cons. But there are also other advantages. For example, we can use the full genetic potential, and we can start looking for novelty in the metabolites that we want to genome mine. We can also look for predicted bioactivity. That's a very interesting idea, to be able to look at the BGC and predict, is that an anti-cancer? Is that an antibiotic? Do I want to go and try to isolate antibiotic BGCs with connected metabolites? So that's something we can start exploring. And then also predict substructures and stereochemistry, like Justin mentioned. Sometimes it's very challenging, the isolation structure elucidation. So the structure elucidation can have a lot of aid from the gene cluster, because we can see the substructures and their stereochemistry. So that's a very interesting aspect to have in this kind of idea of connecting metabolomics with genomics. So those are the advantages. So now I'm going to enter in how to use the website and the databases and how to prepare your inputs. But before that, I would like to ask, are there any questions? If you have any questions, you can unmute yourself or you can post the question on the chat. I don't have, let me see how I get the chat right now. Okay. Yeah, I have it open here. Or we can keep going and then you can ask the questions later as well. It's not a problem.
Maybe I can ask something just to clear it out.
Sure.
So you showed, and thanks for the presentation. So you showed an example of this really big molecule that comes from a known biostatic gene cluster, orphamide, yes.
Mm-hmm.
And could NPOmix link a compound with an unknown biostatic gene cluster, like an unannotated one from anti-smash?
Yes, great question. We do have an example for this biosynthetic gene cluster here, palmaramide A. So we know the structure, the molecule, and we know the MS metabolite. We did the isolation, the structure elucidation, and we had the genome. And then we connected automatically to this putative biosynthetic gene cluster. And then by looking at the monomers, we're able to confirm that most of the structures match perfectly. Some parts that are in red are difficult to predict because they are hydroxy acids instead of amino acids. So they're a little bit more challenging for NSMASH to predict. But yes, we can. Because actually the tool does not know that it's palmaramide A or that the gene cluster is orphamide C. So it doesn't know the annotations. So it makes the connection regardless of the annotations.
So it only knows the domains.
Yeah, actually it's the similarity.
Just the families.
Just the similarity. Yeah, the domain similarity between which makes the families. Yes. Mm-hmm. And in the case of the mass spec, it's actually the cosine similarity, which is the peaks in the mass spec, the fragments.
Great, thank you.
Excellent question, thank you. All right, let me show you a little bit more on how you can submit your samples. So first, the website is available online. Here's the link. I'm gonna send you the slides so you can click in the link. We have a preprint that explains a little bit more. Unfortunately, I couldn't go too much into the details of NPOmix, but the preprint does, and we are trying to publish the paper. We're working on the reviews now, and we're gonna send it back soon. So, and then there's also a Python version. So if you don't want to submit your samples online, if you want to run the samples yourself, you can download the Python version. But I recognize that in the metabolomics community, genomics as well, sometimes we don't have a lot of expertise on the coding, which is not a problem. We can use these tools through this online application that you can just go into the website, just put your name, your email, a brief description like marine cyanobacteria with anti-cancer potential, something like that. And then you can include in 3 lines the link for the FASTA genomes. So the genomes in FASTA format, and then the link for the raw LCMS files. It can be MZML or MZXML. So not raw, not the vendor file, but already converted. And then also the link for the molecules, the MGF file. So you can use that. I'm going to show you a link explaining a little bit more. There's a pretty nice workshop on how to use MZMine to get this MGF file. So just here again, just saying that just a few things that if it's challenging to run in Python, you can submit your samples online. And then if they're already public, we would like to start a collaboration. If they're private, we would like to ask to become part of our private database. So we would like to keep it private, the genomes and metabolomes, but we can publish things like 16S and discovery compounds. And there's no need to share authorship, so no collaboration is necessary. We'll give you the results, and then you can use the result as the way you want. And then again, the files that are needed. So here again, just
Oh, so Tiago, quick question. So then basically, if we submit our samples to you, This is just you'll run the workflow on our samples as opposed to us just running the workflow on our samples.
Correct. Exactly. Yeah. And then you can select if it's gonna be, if you're saying you want to keep your samples public or private,
Okay.
right? You can select.
Okay.
Yeah.
Okay. Cool. So
Yeah.
this
So
is
if
for
I
the
may,
us lazy
I-
ones, I guess.
I'm sorry.
yeah, so if I may add, so even though if they are not public yet and you want to run it and you send it to Tiago, then I still encourage you to include Tiago in the authors, right? In the author list or in the whole publication, because, well, it will hopefully be a nice part of your work. So just to add that. Yeah.
Thank you, Justin. Yeah, so we'll run the samples for you. There's only one downside that I mentioned in here, that we keep limited. For now, we're limited to 50 MS spectra, but I already submitted a grant proposal that it's being scored, and probably we will get the results soon. And if we get the grant, we'll be able to buy a server, and then we can run thousands of spectra. So it'll be much better. But for now, we are limiting only to 50. But depending on the volume of samples that we're collaborating with people, we might be able to run more. I was able to run very quickly in 3 to 4 hours, 550 MS spectra. So that's in one day. So I can run that with no problem right now, because the volume of collaborations is not super high right now. But later on, we have a server, and then we can run many more, just like NTSmash does and GMPS does as well. So here, again, the 3 types of files that you're gonna need: the FASTA files, and here is just a nomenclature thing. Well, we can always correct that, but it's nice if you already send with the proper nomenclature. So you have a name that you gave here. I'm using here an NCBI code. So it just makes things a little bit more simple, more organized, because the NCBI code you can just access and see what is the actual bacterium or organism. And then, so 2 NCBI codes here. And then you have here, for example, one of them only have this one only have a single LCMS file. That's fine. There's no problem. Maybe we only did 1 crude extract in here. And here, the second one, we might have done 3 different conditions. So we have 3 different files. So all we have to do is to add 1, 2, 3 to the end of the name, and that's it. Then you can use those MZXML or MZML files to run MZMine, and then you generate those 2 files in here. We're only gonna need the MGF one, and that's what you submit, those 3 the 3 types of files. You can use a Dropbox or a Google Drive link to submit the FASTA files, and then the LCMS files. It can be separate, it can be together, it doesn't matter that much. So one thing I want to emphasize, if your samples are not public yet, but you want to make them public, please submit to the Paradomics data platform. Like Justin said, is an excellent initiative to help people to develop new genome mining softwares. So it's pretty simple. You have the metabolomics information with you need to submit also to GMPS or metabolites, and then you fill out this information. You have the genomes, the metagenomes or genomes, it can be either, and then you submit the information. For such, you need to use GenBank to submit the genomes first. So GenBank, you're gonna need a bioproject and a biosample, and then you fill out the metadata, and then you submit to GenBank. It's not that challenging. And the LCMS data, you're gonna need, like I said, GNPS massive submission. So again, you enter massive, and then just this simple screen with some details, description, and you submit the files. Here you submit the files, and that's it. So I really suggest that if you want to make your samples public, submit to the Paradomics, and consequently, you need to submit to GNPS and GenBank. So any questions in this part so far? Go ahead.
Yes. So one question. You said that you need these the MGF file, sorry, that's just the MS2 spectra.
Yes.
So does it have to be from MZMine? Because I mean, I think, so I'm working with OpenMS, for example, and it would be really nice if I could still use that and not have to like go through MZMine and just a new software.
No, that's
And
yeah, no, it doesn't. It doesn't. All it has to be is a file with the MGF spectra. So it has
okay,
like a beginning ion and then the information of the ion. Let me open a quick
great.
file.
I think you can do that also from ProtoWizard, no?
Yes, I think so. I think so. Any tool
Ah, good.
that can generate that kind of file, you can definitely use. There's no problem at all. Let me see if
Because I think from ProtoWizard MS Convert, you just convert the MZML to an MGF. I haven't used it, but I think it's possible.
let me see if I can find 1 second. Yeah, I just want to show a quick MGF. Yeah, a quick MGF file. Oops, no. Because 1 second, because the bar is in front of that's here. That's right here. So yeah, so you just need to have a file that it's an MGF. It can be, I'll show you, you can use individual spectra. They can be separate, no problem. Or they can be in a single file, whatever the tool generates for you. So no problem, you can submit either way. So all it has to have is this beginning ion, and then peptide mass, and then some other information. Some of them is not required. You don't need to have a SMILES or a data collector. And then you have the spectrum ID is important, and then the ions. So the ions and the intensity. So those are the fragments. And then this is pretty large one, but it can be smaller, and ion at the end of the file. So this is only showing one, but you can have several together, or you can have, like I have in here, in one folder, several separate. So either or works.
Great. Great, thank you.
No problem. That's an excellent question. Yeah, I forgot to mention that. Thank you. So let me just show briefly how to get those MGF files. Just as a suggestion, like we just talked about, you can use different tools, but I'm just suggesting some ways to do that. There are many ways to do that. So the first way to do that, I'm showing the example here, and I'll show the results for this example, is Jamaicamide A. So here you have BGC from MIBig, like Justin explains, a database for biosynthetic gene clusters. It's nice to say that is an expert annotated database. And then, so you have this information about Jamaicamide, and it's connected to this structure. And we know that because Paradomics also has that information, we also have it connected to MS spectrum. But how can we get this MS spectrum? Well, we can go to this website here, which is a link in GMPS database. We can search for Jamaicamide A here. So Jamaicamide, then you can click in the metabolite. It's gonna open its page. So you see a link to MIBig, you see a link to Natural Product Atlas, which is another database of natural products. And then you can click download Spectrum Peak. That's it. You downloaded the MGF. So you have an MGF that you can run in NPOmix. Of course, it's a known MGF. So if you want to run your molecules, I'll show you in a minute how to do for unknown MGFs that you have in your sample. So again, another example I'm going to show the results is cryptomaldamide. Same thing. We do, here is the structure. It's on MIBig database. We do the same process as Jamaicamide, and we get the MS spectrum. Okay, what if it's not in GNPS? I don't have the MS spectrum. I just know that I have the gene cluster. I see that the gene cluster is in my bacterium. I know the structure. I know the M over Z, the predicted M over Z. How can I get this MGF file? Well, we can use this workshop I'll also put in the website, the NPOmix website, the link for the workshop. It's actually already there, but I'll make a extra pop-up with the video that you can use MZMine, but again, we can use other tools as well. But MZMine generates this MGF file for your samples. So you use this MGF file. You use your samples in MZMine, the ML or MZXML files. You use them to process them, and then MZMine generates this MGF file. Then you can split this MGF file in several files, like I showed in that folder. I was showing you the MGF file. You can do that, and then you can search the mass for this example, Brasilia-Cardin A. So then it points out that it's this MGF file, and it is also present in the concatenated version, of course. But then you can just separate that, and now we have 3 different MGF files for 3 known metabolites. That 2 of them are published. The MS spectrum, the spectra are known and published in GMPS. One of them is not. So we just have a putative possible Brasilia-Cardin A. So there's a tip. There's another way to do that, and that probably applies to many other tools, which you can import your data. Then once you have your file here, the MZXML or MZML, you can click on it, select the range or auto range, and then click OK. And then you're going to see the MS1 spectrum. And then you can click in a particular peak, let's say it's this peak. And now you can see all the MS1 in that peak, and then you can select a MS2 to load, and now you see the MS2 spectrum. And with this quick button here, you can save the MGF file. So I'm just saying here, sorry, I didn't translate from Portuguese. I'll do that in the last version of the slides. But I'm just saying that we have a list of names that you can use to include, because it's necessary for now for NPOmix to run to have a CCMS ID. So that's something we're going to change in the future. You can use that list to name your MGF file, or we're going to do that for you. So it's not an issue. You don't have to worry about that. You could do it, but it's not a problem if you don't do it. So yeah, so that's how you get the MGF files. So now we have all the MGF files. You already had the FASTA. You already had the LCMS MZXML or MZML files, and you can just submit in the website. And then we're going to run for you, and then we're going to show you the outputs. And I'm going to show how to explore the outputs very briefly. But is there any question about the input data?
Chaguinho, I do have a question.
Hey, Mauricio, how you doing?
Good. Sorry, I'm late.
No worries.
Tiago, so at the beginning you say like, oh, you have the genome file, and it can be a FASTA file. Sometimes when people get it from the NCBI directly, you can download it. It can take time, but you have a format of .fna, which is basically the same FASTA. Do you have an issue with either the name of the file, like .fasta or .fna?
No,
Because it's basically the same.
Yeah.
Okay, perfect.
Yeah.
And
just rename it to .fasta, and or if you don't, we can do that for you. It's not a problem. And it's the same actually, because we're gonna run NSmash. NSmash uses both, so it accepts as input either FNA or FASTA, so it's not an issue. Yeah.
Perfect. And the other option is like automatically people need to download the FASTA file, or you just provide the link to the NCBI directly for the workflow to run it by itself without the person to actually download the file?
Yeah, either or, it's fine. You can select. But some people don't have the FASTA yet published at NCBI, so they can just submit their files. So it's not a problem. But if you have everything is published, you just give me the NCBI code, and then I can download that in seconds. It's not an issue. It's pretty simple to do as well.
Perfect. Makes sense. Thanks again.
Yeah, actually, Paradomics, you have all that information for more than thousands, I think 1,700 samples that you have all the FASTA files, the LCMS files, you can download all from there. I haven't been able to download the full database because some of them are JGI links instead of NCBI links. And JGI links are a little bit more challenging to download because it kind of needs to do one by one. So it's, yeah, you need to get an email and then you get the link.
Yeah.
So to do like 700, it's very challenging.
No, yeah, of course.
Yeah, but I'll try to contact them and see if they can help me.
Sounds good. Thanks, Eugenio.
No problem. Great question. Yeah, let's take a quick look on the results. So again, here's the website. You can go there, submit your samples. We're gonna run. You're not gonna see this screen. This is for us in the back end that we're gonna run. It's a very simple way to run. I actually will have, I do have a video already in the website on the Python version, but I will also provide a workshop on the Python version, how to run it. But you don't have to do that. You can just submit your samples and we will run for you. But this is what happens. You just run Python npomics.py. We already put all the locations for the files and it's gonna run. This is the longer part of the process is the cosine comparison. So comparing the MGF files to all the LCMS files in the database. Right now we have 3,300 LCMS files. That can take a while. So that's the one that takes the longest. So for example, 3 examples, it took 1 hour and 25 minutes. But for 500 and actually 550 examples, it took around 4 hours. So it can be pretty fast. And I want to remember that this is all being done so far in a MacBook. So in a regular laptop, only 8 cores, I think 8 of memory as well. So it's not a potent computer. So when we start using a server, then we're gonna be able to run many more samples and in a much lesser time. So yeah, so for now we're limiting only 50 MSMS spectra, the MGF part. But again, you can use the Python version and there's no limit that we know of. The maximum we tried, it was 1,200 samples and it ran pretty well. It took much longer because they were very large metabolites. So that's important also to mention. It was 550 that it took 4 hours. But if we have 550 metabolites that are much larger, then it's going to take a little bit longer to calculate the cosine values. And this is the output, the outputs. We have here 6 outputs. The first one is just a list with all the BGCs. So very simple here, this list. So here is a genome, and then here's the number of the BGC. Smash uses this nomenclature, region 18, region 2. So yeah, so the different ones. And then you're gonna see some start with BGC. Those are from MIB. So those are validated non-BGCs. Then the second output is this dictionary. Actually, the second and the third are very similar. So they are this dictionary. So it's a lot of stuff in the screen, but it basically says GCF1. So the first gene cluster family, and here are the BGCs in this family. So that's all it's saying. So for example, in GCF1, there's no MI-big BGC. There's no BGC starting with BGC00 something. In GCF2, there are actually 3 MI-big BGCs. So most likely GCF1 is new and GCF2 it's not. So the second dictionary is very, very similar, but we use the NCBI names instead of the names that were originally in the samples. So that's the only difference. Then we have the testing and the training matrix. So very briefly, the training matrix you have here. So 0 from the number of BGCs you have, those are in the same order as this list here. 0 from 15, for example, those are the same 0 from 15 here. And here in the columns, we have the samples. So we see that BGC 7 is in the this sample from column E. And it's present there, there's something similar that is about 90%, 89% similar. So that's how to interpret the training matrix. So NPOmix is gonna use this matrix to train in the BGCs, plot them in the KNN space, and then use this testing matrix. We only have 3 examples, so only 3 rows in here. So the same, we're gonna have the same number of samples in the columns. So here is 1,040 samples. And then you see the same idea. This third one is present in the column C in 93.5% similarity. Okay, so the final result is the most important, is the actual connection. So you have a metabolite in the first column, and then you have predicted BGCs. Here we use 3 nearest neighbors. So 3 possibilities, so 3 candidates. And then we have some extra information. I'm gonna talk about max Jacquard index in a little bit. You have the parent mass, and then you have the peak count. So this is the peak count above a certain threshold of intensity. So we can call intense peak count. So here there are the 3 metabolites that we use. So Jamaicamide, Brazilocardin A, and cryptomaldamide. Let's start with Jamaicamide. So Jamaicamide, like I said, is in NCBI, in MIB, and the code is this one. And then when we look at the first candidate, GCF471, this BGC for MIB, it's present in GCF471. So it indicates that, okay, we know what is this metabolite, and it is Jamaicamide A. And that's correct, because this metabolite was downloaded from GMPS, and it is Jamaicamide A. The same can be done, for example, for the third row, we have here Brazilocardin A, and this is the MIB code. And again, we see that the code is in here. So this GCF456, it's definitely known, and it is Brazilocardin A, which is really nice because we didn't have the MS spectrum for Brazilocardin A before. Now we do, we name it A1213, and now we know that it's connected automatically to the BGC. So we can actually publish this new MS spectrum at GMPS as a level bronze, so that we have an indication that it is the Brazilocardin A, but we haven't confirmed with, for example, purchasing a standard and confirming this structure. Actually, although it was done by not collaborators, but another group that published that in the NPOmix data platform. So this is a validated link. So even though we didn't know the MS spectrum before because it wasn't in GMPS, we have an indication of the retention time in the mass in the NPOmix, and that data was validated. So we know now we have a validated MS spectrum for Brazilocardin A that we will publish in GMPS soon. Okay, just talk briefly about the Jacquard index. What is the Jacquard index? Well, we have here 1,040 genomes or metagenomes, and actually it's a combination of both. About half of them are metagenomes, the other half is genomes. So that's another point, that it works for both genomes or metagenomes. And here we have the maximum similarity score, those scores that we're looking here, for example, in the training table. So those scores in here, they are the same that we have in here. And you can see in the metabolome, it's the same idea. We have the metabolomes, so which means the LCMS data. And then we only have this one single score in the last sample over here. So it's not matching very well the scores in the left side. So this is a low Jacquard index. When we compare the presence absence of those scores, it's gonna be a low value. What's a high Jacquard index? You can see that here it's only present in one sample, and it matches perfectly the same sample in the metabolite sites. Here we have 2 gene clusters. They are superposed in this same sample. And here we have 5 metabolites that are also superposed in the same sample. They're all albocytins. So they're matching the albocytin BGC. So it's a correct match And then consequently, the Jacker score is going to be really high. For example, 1, because it's matching perfectly. But what if the metabolite, what if we know the metabolite, like cryptomaldamide, but we don't know the BGC? So let's pretend this MI-big BGC is not here. What we can do is we can pick one. It can be either this first one or the third one in here, either or, and then we can open the NSMASH file. The first thing we see is aminotransferase. That's a pretty rare gene that makes this portion of the molecule. So that's kind of a nice confirmation that most likely this BGC that was automatically connected using NPOmix to the second candidate. The first candidate was Jamaicamide. The second candidate was actually the cryptomaldamide gene family. This kind of confirms this part of the structure. Then the first amino acid is unknown, but we know it's an amino acid, so we know a little bit about it. The first amino acid is cysteine, but we are predicting to be a serine. So there's a difference, but it's still amino acid. It's pretty similar. The second amino acid we don't know, which is valine in here in the structure, but the amino acid was not predicted by NSMASH. But we know it's an amino acid, so we know this part. The third amino acid matches pretty nice. It's an N-methylvaline, so it's not a very common because of the N-methylation, is not super common. And then we have a PKS extension that we have in here. So most of the structure matches the predictions from NSMASH. So that gives us an extra confidence that this link made by NPOmix is probably correct. So that's how you can investigate unknown BGCs. I also show a better example where we have several amino acids matching. And then I also showed you another example where the gene cluster was not known, but the structure was known and the MSMS spectrum was known. And we connected, and by looking at the biosynthesis, we can kind of confirm as well the match between the gene cluster and the structure and the metabolite itself with the MSMS spectrum. And then there's an extra thing that I would like to emphasize. Okay, what if we don't know the structure? What if it's a completely new molecule? We connect to it could be a known, it could be an unknown BGC, but it has these predictions of amino acids. You can look at the mass differences between the fragments and then start to make sense of them. Maybe looking for maybe 128 is isoleucine. I'm not sure, but just saying that it could be. Then we can start making assumptions that maybe this molecule has isoleucine, just like it's predicted in the gene cluster. So those are the kind of extra steps you can take to look at the molecules like cryptomaldamide or orphamide C or palmaramide A to confirm the links between them, or even new molecules that we don't have examples yet, but we're working on making those connections right now. Any questions?
One question. Thank you, Thiago, for the nice presentation. I have a question regarding maybe if you have one example when you're able to connect like unknown BGCs to known structures, like that the BGC it was initially identified, but there are no natural products associated with it that haven't been isolated from this BGC.
I'm not sure I got your question, sorry. So if you have a new sample that you have BGCs that are not known, so you don't know what they're making, or they're known?
They are identified as BGCs, but they are not known like what they can produce.
Yeah.
Are NPOmix able to link like the unknowns between them, like unknown BGC with unknown MSMS data?
Yes, there's no restriction because actually NPOmix doesn't know if the BGC is known, unknown, if the molecule is known, unknown. So it can connect any of them. So it can do either or. In the version 2.0 that I'm going to show you right now, we do need to know some extra aspects of the samples. So for example, some unknown metabolites we might not be able to predict what class they are. Is that a peptide? Is that a polyketide? Is that a terpene? So that's for version 2.0. But version 1.0 only uses the similarities. So it just compares them. So it could be known, unknown. It doesn't need to be known to be able to connect, which is really interesting because, well, we actually want to go after the unknown ones, right? We don't want to, I mean, dereplication is important, so we don't keep isolating known things. That's important. But more importantly is to go after the unknowns.
Thank you.
Okay, so just a little bit more, just a few more slides. Just quickly introducing you the second version of NPOmix. So the first version that I talk about today, we use similarity fingerprints. So those similarity scores, and then we need the label because it's a supervised approach. So we need the label to be able to train. But what we thought was, okay, can we add, for example, biosynthetic class, the presence absence of the class? Is it a terpene? Is it a PKS? Is it an NRPS? Is it a RIP? So can we add that? Would that improve the training? That's the question that we have. Also, how about substructures? Does it has a tyrosine, a proline, an amine, amine, a malonyl-CoA, carrier reductase domain? So can we introduce those substructures, kind of monomers, into the information in the training set, and would that improve the training? And the answer is yes, it does improve the training. So here's a precision curve where you have precision here, and then in the x-axis, you have the K value. So the number of neighbors, the number of candidates. So here before, I show you 3 predicted candidates. So the K was equal 3. Here now, I'm using 1, 3, 5, 10, and so on and so forth. So of course, the bigger the value of K, the easier it is to make a prediction that is correct. If you're predicting 10 candidates, the chances of the correct one being one of them is much higher than just one, for example. Here in the blue is the version 1.0. So K1 is only 12.5%. K5 is 50%. So 4 times improvement, for example. So we probably want to use something like K equal 3. That's what we decided to be the best. So looking at the improvement, so here we have blue is the version 1.0, green is the version 2.0 with classes and similarity. Blue is only similarity. So brown is version 2.0. So we don't use the class, but we use substructures and similarity. And then pink, it's all 3 of them: similarity, substructures, and classes together. So what we can see is that for K equal 3, and then it's important, we are not using a Jaccard filter. Once again, Jaccard is to check the equivalence between the genomes and the metabolomes if the metabolite is present in the same samples as the BGCs. So like I showed you here before. So we can use a filter saying we only want matches that have 0.7 or bigger Jaccard index. That's the Jaccard filter. But here we're not using the filter first. And we see values like version 1.0 is 50% precision, which is okay, but it's not very good. We want a higher precision. And with no filter, we can go up to 77.3% precision using either the substructures and similarity, or substructures, similarity, and classes together. So a pretty good improvement of 27%. So that's an indication that we should use these new features, the class and the substructures, to make our predictions. Then actually I want to say classes are So looking at the filter, classes are even more appealing because, for example, with Jacker filter, the precision scores improve a lot, which is nice. So it's nice. I always recommend to go after hits that have very high Jacker index. And then what you can see is that from version 1.0, we have 78%. It goes to 93% precision using only the class. Why is this so appealing? Because right now, it's kind of challenging to predict substructures from MS data. We're trying to use MassQL, which is a new tool that uses a query language for mass spectrometry to be able to predict those substructures, but it's kind of challenging. But with Canopus or MS2LDA, we can predict pretty well classes from MS data, straight from MS spectra. So it's nice to see that we achieve precisions like 93%, only using similarity and class that we can infer already from MS spectra. So just some limitations. Of course, you need first the fragmentation data. If your LCMS only has MS1, unfortunately, we cannot use NPOmix. Also, it's important that BGCs in the genomes, if they are not similar to anything in the data set, what we call orphan BGCs, they cannot be annotated by NPOmix. So that's an unfortunate limitation that we have. So you need at least one other sample that is similar to yours. So the BGCs network make families, and then we can use NPOmix. And if the BGCs are not expressed in some or maybe all strains, then it's also very difficult to connect to a metabolite because the metabolite won't be there most of the time. So I just want to highlight NPOmix is a prediction tool and it generates false positives. As you can see here, thankfully, the precision is pretty high, so the number of false positives probably is not that high. It's a pretty low number of false positives, but it's important to mention that we do generate false positives, and we're trying to improve the algorithm to increase the precision, and it's important to select high Jacker index. So next steps, we're expanding the database. We want to include more fungi, algae, and plants. So far, most of the samples are from bacteria, which is not that big of a problem because what if the BGC that you're interested on from fungi is also present in the bacteria? Then we'll be able to run NPOmix. So it's not that big of a problem. I still advise you to test your samples even if they're not bacteria. We want to create a better interface for exploration of the results, and we want to buy a server like I mentioned before. So again, here's the website. You can submit your samples there. You can see more details on the tool there. There are some videos explaining more details on how it works. You have the preprint. Hopefully soon we're gonna have also the publication. So just to finish, I would like to compare very briefly other multi-omics tools. Like I mentioned them, I mentioned those 9 tools, including NPOmix. And we have a benchmarking data set with 4 NRPSs, 3 PKSs, 11 PKS NRPSs, 1 terpene saccharide, and 3 other, so none of those canonical classes. And that's a total of 22 MS spectrum and metabolites. So we try to test with all these different tools. And here is the link. So if you're interested in using these tools, and I highly advise you to also give it a try in your data and test those different tools. So some tools like NPOmix have the website version and the command line version. Some other tools have only the command line or only the website. And some cases, unfortunately, some of them are not available anymore. So that's something that I struggle. It could be that I'm going to the wrong website. So if you find the correct website, please email me the correct website. But this is what I found. For example, GMP here's in the McGarvey lab, it says that the service is unavailable. And I could not find the command line version. Some other problems, for example, DipRip, I was not able to load the FASTA files But those FASTA files ran with no problem with NSmash, NERPA, MetaMiner. So it's kind of, I don't know exactly what's going on with deep-reap in this case. And I wasn't able to find the command line tool either. MetaMiner, it ran, but as you can see, we don't have any rips in the benchmarking data set that we have. So I'll comment about that in a second. Well, actually, I can say already. So what I tried to do, I saw that in the genomes of the strains that we were using for benchmarking, we have 49 rips. So I tried to use MetaMiner and deep-reap to connect them to unknown, so new MS spectra. But I got 0 hits. So none of those gene clusters, RIPS gene clusters, were connected to any metabolite. NRP Quest seems to be not available, so we entered the website. There are other tools, but NRP Quest is not there. I couldn't create an account using NRP Miner, but my collaborator has the code for running offline, so he's running right now for me. NERPA is available here. It's very simple to use. You can download, you can run. It's a command line version tool, but it's very simple to use. Garlic, I tried to run it, but I wasn't able to compile the tool. So if you know how to compile that, please send me an email as well with a quick suggestion on how to compile this tool. I really would like to test Garlic in my samples. So out of the 9 tools, actually 3 of them seems to not be available anymore. And then 3 of them I wasn't able to run with my data set. It doesn't mean that they don't work. It just means that for this particular data set, I wasn't able to run them. And it could be that I'm going to the wrong website. It could be that I'm using the wrong command. But so far, I wasn't able to run them. So NP Linker, I already run the test version, and it worked really well. So now I'm trying to run this particular data set as well. NRP Miner, my collaborator, is running for me. So I think I can say that NPOmix is systematic, high throughput. It's pretty fast because it works in a MacBook for a pretty large number of sample. I mean, not that large for machine learning, but large enough for getting good results. And it's highly efficient, 93% precision so far. So it's pretty good. But I would like to mention that NP Linker is also systematic. It's also high throughput. And I'm still testing if it, for this particular data set, how to compare in terms of speed with NPOmix. So I cannot make any conclusions right now. Also, the same thing with efficiency. I'm still testing that. But it could be that it's also high speed and high efficiency. So NP Linker is definitely another tool that you should check it out. And like I said before, there is definitely room for, there's a lot of overlap and then room for synergy between NP Linker and NPOmix. And that's what we're gonna do in future. We're collaborating right now, so that's very promising. And that's we're working together to make a good solution, probably more a unified solution as possible to this genome mining problem. So I'm trying to create this graph, but unfortunately it's not complete. I still need NP Linker and NP Miner, and NERPA didn't generate any result. So I ran, it could link 11 metabolites, but it did not link any of the metabolites. So I couldn't tell the correct or incorrect links because none of them were linked, but it should have linked those. And then those other tools, Garlic, NRP Quest, GMP, MetaMiner, and deep-reap, I cannot use them and generated no results as well. So this graph, once it's completed, will be in the publication for NPOmix. So I just want to talk just this last slide. I just want to, because we have 10 more minutes and we can use some questions as well. This last slide, it's kind of nice to see the background from people. Most people did not use any of the tools until now. And I kind of reckon why, because like I said, like here, some of them are not available anymore, or if they are, it's hard to find them, or it's hard to use them, or they yield no results. But we want to change this picture. We want to make NPLinker, NPOmix, and hopefully have also workshops comparing and using all their tools to make them more accessible, more easy to use, to provide the proper instructions that in some cases can be missing. And we want to change this picture to have more people using those tools. And I was surprised to see that GMP was heavily used. I could be going to the wrong website, but I believe, well, I cannot see, one second. Cannot see the bar. That this is the McGarvey Lab website. And then here, GMP, that's what I did. It's not clicking there anymore. One second. So I click in GMP and then it says service unavailable. So I'm surprised a lot of people use the tool, but unfortunately right now it seems to be unavailable. But again, if you know and you have suggestions on any of these tools and how to use them or how to fix the bugs, and please contact us. Or if you want to share right now, that's the time as well to share this kind of suggestions, and yeah, we're open to hear your opinions. Thank you, guys.
Awesome, checking you. Really good.
Thank you. So if
Thanks,
you have any questions.
Could I add that for NPLinker, there is a Docker version available. So that should be, in theory, quite convenient to use. There's also command line available. So instructions are on the GitHub.
Yeah,
and please, with issues, if you have them.
Yeah. I think I put the correct. I think I put the correct GitHub link in here, so you can click.
Currently located there. It will change at some point, but then that will be clear from that website as well. So
yeah, and the instructions are really clear, and the Docker version is really easy to use. I had some bugs that I'm working with Andrew, and he answers really quickly, and he's very helpful. So NPLinker is a really nice tool to use, and I strongly advise to use both NPOmix and NPLinker and other tools as well. But unfortunately, the experience I had so far was much better with NPOmix and NPLinker, to be honest. We have a question in the chat. So it says, is the purpose of using MGF files for target studies of known/unknown metabolites in the sample, as we need to extract them manually? Well, it doesn't need to be target studies. It's just that simplifies our pipeline to have already the MGF files. Because what happens is in NPOmix, we compare the MGF files with the MZXML files. So we need to know which files we want to compare. So what we could do, we could extract the files for you from your on-target MZXML file, MZML file. But it will help us a lot if you already do that step, extract them, and then say, I want to investigate those files. Also, because maybe you're only interested, let's say, in NRPSs. And then what you can do, you can run Canopus and say like, oh, out of the 1,000 molecules I have in my data set, only 200 are in NRPSs, and I want to focus on them. So you can give us only the NRPS, the 200 NRPSs, NRP, in that case, non-ribosomal peptides, in your samples, instead of all the 1,000 MGF files that you could extract from your sample. The other thing is limit of detection, because if we use, let's say, a threshold of 1e⁶, it's a pretty high threshold. You may only get the major peaks, and you might generate, let's say, 100 MGF files. But then if you want to be more less strict, you can use 10 to the e to the 4, sorry, 1 e to the 4, and then you can get like thousands of MGF files. So it's going to depend on your preference. So that's why we ask you to be able to extract the files yourself.
Hey, Tiago, mind if I ask a question?
Sure, yeah, go for it.
Hey, great talk and a great tool. I was curious, I noticed that you have anti-smash outputs and Bigscape outputs are one of the things that you're generating there. So are you running anti-smash and Bigscape in the background using the user's input?
Yes, definitely, yeah. Yeah, those are
Okay.
actually the longer steps because, I mean, for field samples, NSmash is pretty quick. We can use the online version for field samples. It's pretty fast. But if you have hundreds of samples, then we got to use the offline version, the command line version. And then we need a server to be able to do that. And then it does kind of fast, but it takes a while. And then Bigscape I'm running on my computer. Right now I have about 1,050 samples. We got 10 extra samples. So if you add your, let's say 100 more samples, then we're gonna have all those samples to run Bigscape. That might take about 14 hours in my computer, my MacBook. So of course, if I'm using a server, then it's gonna take like 1-2 hours. So it's much faster. But right now I'm just running my MacBook and it takes about 14 hours. And then NPOmix takes like, if it's only 500, 550 MGFs, it takes only 4 hours. So it actually takes longer with NSmash and Bigscape than NPOmix itself. But we need to run them; it's necessary. So it's part of the tool as well.
As a follow-up, if a user has already generated those data sets, is there a way for them to input those into your tool? I think that's kind of how NP-Linker does it.
Yeah, we can always. So if you do have that, please on the description when you're sending your links, mention that you already have the Bigscape or the NSmash files. That will help a lot. That will definitely help. And then we can input them straight to the tool. But
okay.
That's actually something I will start mentioning in the future. That's a great comment.
Okay, cool. Thank you. I appreciate all of the workshop. Thanks.
My pleasure. I'm saying that Pedro also has a question here. Thanks for the talk. Thank you for listening. So once the bottleneck for on-target metabolomics is mainly the annotation process and thus the identification of these metabolites in its classes, do you suggest apply NPOmix in this context for peer metabolomics study? Yes, so that's the idea that so annotation is definitely the most difficult part. Sometimes we can only annotate 5 to 10% of the metabolites in a sample, sometimes more. I think with Canopus, we can annotate many more classes, but not actually the metabolite itself. So connecting with genomics can help because you get so many other annotations from the BGCs that can be very helpful on things like deciding which metabolite to prioritize. That's a major question. If you have a sample and then you use a pretty strict threshold, but you still have 1,000 metabolites, which you can only isolate 1 or 2. So which one you're going to prioritize? So start annotating them definitely helps to prioritize them and be like, okay, I want to focus on the RIPs that are connected. So the metabolites that are connected to RIPs BGCs, because I have structured predictions from them, and also the antibiotic ones, and also the ones that are present only in 5 samples out of 1,000. So those kind of extra bits of information, they can really be helpful on the decision of like, okay, well, out of this 1,000 metabolites, which one I'm going to isolate. So I think this connection between metabolomics and genomics really helps in this process, in this annotation process, where you can start making more links and not only links between them, genomics and metabolomics, but links between pieces of information like antibiotic activity or anti-cancer activity or a certain class of BGC, or maybe a BGC that is only present in Streptomyces or Salmonespora. So yeah, so those are important kinds of information. Thank you for your question, Pedro. Oh, when you say pure metabolomics studies, uh, do you mean like without using genomics? Oh, okay. No, we still, well, we're thinking about this. We still thinking, okay, it could be that a metabolite it's present in a particular sample, but the BGC is present in another sample. That's totally possible. Actually, right now, the tool does make those connections between a metabolite from one sample to a BGC to another sample. We don't like those connections as much because we prefer the connections that are from the same sample. Because but it can happen because it could be that a BGC from the sample that the metabolites found is degraded. It's fragmented in several parts, but in another sample, it's not. So we can make this cross sample connection. That's definitely possible. And in that case, it would imply that you don't need the genome from one sample to still connect a metabolite from one sample to a BGC to another sample. So it's possible. And vice versa is also possible that we don't need the metabolome and we can still connect metabolites from a database, for example, like GMPS, to samples like genomes from the database that NPOmix currently have. So it's definitely possible, but we're still working on it. There are some challenges that we need to face first, and then probably right now we're working version 2.0, but that's probably version 3.0 that we're gonna offer this kind of service that you don't need the paired data, but you can still run NPOmix. Yeah, great question.
One question, Tiago. Have you think about including proteomics data into this pipeline?
Ah, great
Because,
question.
because I will say that it will improve even more these scores, since when you have absence or presence of some BGC, then it would reduce the cases for most of the links.
Yes, I was thinking about transcriptomics first. Start with transcriptomics because then we can look at because we're making sort of correlations, right? It's a machine learning approach, but it is still making kind of a correlation based score. So what I was thinking was if we know that a BGC is inactive, then we can remove that from the correlation. So then we can get better links, and we can get links that are more likely to be correct. So that's definitely either transcriptomics or proteomics could definitely help with that, and that's something I want to add as well as an option to this. That'll be a very simple addition to the pipeline, and you probably improved a lot. Because it's also a curiosity, like how many of those are actually active? How many are actually making the metabolites, right? Like that's we don't know for sure. Like I've seen few papers that make some estimations, but it's a very challenging question to tackle, and then we don't know yet like how many of them are actually active.
Yeah, it's exactly, because sometimes you are trying to link the orphan VGCs to some MS features, but having like all the data without knowing either is active or not the VGC, then it's a whole mess.
Yeah, definitely. Yeah. Yeah, so definitely we're looking into including other omics as well. I mean, the more data we have, that's machine learning. The more data we have, most of the time makes better predictions. So so we think also once we have like a 10 times bigger database, we include transcriptomics, we include other features, it's gonna make the tool better and better. So yeah, so it's a good start right now, but it can only improve. So that's kind of exciting.
Thanks, everyone.
Thank you, Tiago and Justin, for the nice talk.
Thank you.
Welcome. And yeah, have a pleasant day, afternoon, evening. And well, speak soon again.
Bye-bye.
Bye-bye.